One, two.
Good afternoon.
Hi,
Professor.
Nicholas. How are you?
I'm doing good. How you doing?
Fine, thanks.
No
Fine.
problem.
Now lost me again. Forgot how to do this. I can't remember now how to do it. Okay, good afternoon, everybody. I hope everybody can hear me this time. I think so. Today I'm going to try finish photosynthesis, our discussion of photosynthesis, and then we'll hopefully move on to talking about the cell cycle and cell division. Before I do anything else, though, I have to tell you what wonderful grades we got for the midterm. I was very, very impressed. Given all of the difficulties of studying online and all the adaptation, all the problems that people have had to contend with, I think that the results for midterm were really outstanding. And I think there's very few people in the class have any problems getting a good grade at the end of the course. I was very, very pleased. Okay, so let's carry on our discussion about.
Professor?
Yes, sir.
Sorry to interrupt for a quick second.
No, it's fine.
Some of the students are texting me saying that they didn't get the link, the code to join, and they're asking me if they can get the link. They said, I don't know. They're texting me that. Hey, I'll read the exact message. They said that he didn't. Where's the link? I can't find the link. I told him to check the email.
Yes, so they need to check their emails. I think I also posted it to Canvas announcements.
Okay, I'll tell them right
I'm
now.
not 100% sure, but I definitely sent an email to the entire class. And it's very difficult for me now with everything up on the screen. It's very difficult now for me to shut everything down and go back to Zoom to find out.
Good afternoon, Professor.
Hi, how are you?
I'm good. A few people also messaged me, and I sent them the passcode. I mean, not the code, but like the meeting ID. I sent.
Yeah,
yes,
I sent it.
Thank you.
I sent it to the chat.
Okay. Then yes, that's that's all I can suggest. I'm very sorry, but if people could just do me the favor of just somehow posting that meeting ID link, I'd be very grateful. I don't know why people are not getting emails. But I am trying to post everything to the Canvas announcements as well. So I'm afraid I've just got to carry on. Okay, so as I was saying, we're going to try cover the rest of photosynthesis today, but I want to do it slowly because I wanted we're only covering photosynthesis in the broadest of detail, but I wanted to clear exactly what we're talking about. So what we learned last time was that there's a series of reactions which compounds are energized by light from the sun. Let's put it that way. And those light reactions, the net result of the light reactions is actually to produce energy-containing compounds. First of all, a little bit of ATP is made. But much more importantly, the electron carrier NADPH, very, very, very similar to NADH. This is the compound used in plants. NADPH is an electron carrier, and it's carrying electrons from hydrogen which have been highly energized by light from the sun. And all of these compounds are donated to this cycle called the Calvin cycle, which is where the plants actually, or the autotroph, this is used in other things other than plants as well. But the autotroph takes carbon dioxide, simple compound with carbon in it, and reduces carbon dioxide and ultimately ends up producing a sugar. This process is very expensive in terms of energy. It uses up a huge amount of NADPH, as you'll see, and ATP, but it doesn't matter. It can be as expensive as you like because the energy is free. The energy is sunlight. And that's how the process is driven. A set of light reactions fund the energy for these highly endergonic reactions in the Calvin cycle. we'll talk a little bit about the Calvin cycle today. I want to just run through the sequence of events, though, for the light reactions very quickly, just to refresh it in your minds. And I'll remind you, these are the systems which are embedded in membranes. In prokaryotes, there are photosynthetic bacteria where these substances are all embedded in the cell membrane. But in eukaryotes, for instance, in the plants, they are usually embedded in the membranes of organelles. And in most plants, the organelles are the chloroplasts. That is where all of these reactions take place. So you can imagine, this is just schematic. We'll see a semi-realistic representation in a while. But there are two light gathering systems. They consist of a very reactive chlorophyll molecule with electrons which can be boosted to a high energy level. The energy which boosts these electrons is derived from the light falling on the energy gathering system. And we call it, it's actually called an antenna because it consists of many pigments which are excited by the light but pass on the energy ultimately to this central chlorophyll. This chlorophyll, by the way, can also be directly excited by light falling on it. But most of its energy comes from these accessory pigments around it, which get excited by the light and hand the energy on. Electrons in that chlorophyll are boosted to a high level and they actually reduce this substance here, which is a primary acceptor. Never mind exactly what it is, but it is an acceptor. It's an electron acceptor. There's a problem. That is that this chlorophyll molecule here now has been oxidized. It's lost electrons. And it can't remain in that state for very long. It's actually a very powerful oxidizing agent. And it is very strongly electronegative. You can think of it that way. So these electrons that have been now accepted up here have to be immediately replaced. And they're replaced through this process whereby water is split. It is enzymatically split. And the electrons from the hydrogens of water are passed back to the chlorophyll. In the meantime, if you take away these hydrogens from water, you end up liberating oxygen. And that is where the oxygen in our atmosphere comes from. It's all from this process of photosynthesis. Remind you, this is a process we would love to know the intricate details of how those enzymes work. And this would be a great source of energy and everything for us if we could mimic this, which plants just do so easily. Okay, so now we now have this electron acceptor up here has been reduced, but that is an unstable situation, and the electron now undergoes a series of redox reactions again. So this reduced is now oxidized and reduces this substance here. This now is oxidized and reduces the next substance. We again pass our electrons through this electron transport chain. And as they descend, each stage, the electrons are losing energy, and that energy is actually harvested. And it is harvested again, as we saw before, it is harvested to pump protons across a membrane and accumulate both a chemical and a charge differential across the membrane. And we're going to equalize that charge by allowing those protons again to run through ATP synthase, the enzyme that can make ATP. So in this product of this electron transport chain is ATP. This electron has a destination, though. It is being attracted by something which is electron negative. And that is this. It is the next chlorophyll molecule located in the next photosystem. This one's slightly different to this. This is called P700. It's optimum light wavelength. But this P700 has been excited in exactly the same way as the 680 was. It has energy from all of these pigments around it in the antenna, has been passed to it. It may have been hit directly by light itself, and its electrons have been boosted up and grabbed by another acceptor. Those electrons have to be replaced. As long as this is in an oxidized state, it's very strongly electron negative, and that is what pulls these electrons through this electron transport chain. These electrons boosted to this acceptor, they have a different fate. They are actually going to be used to manufacture NADPH. And once NADP plus is the terminal electron acceptor for this system. So these electrons, they are emerging, and the energy in them is being used now to reduce NADP plus to NADPH. And this is the substance we really got to watch, because this is going to do a lot of work for us in the next stage of photosynthesis. So at the end of it, everything's back to where it started. This has had its electrons replaced. Those electrons have been through here. They've replaced the electrons that were lost when this was oxidized, and those electrons have been used to reduce NADP plus, and we end up with NADPH, the electron carrier, carrying highly energized electrons derived from hydrogen. So this is a kind of semi-realistic view of how the systems are embedded into the membranes. This would be in the membranes of thylakoids of a chloroplast. And you can see the two photosystems. There's the one photosystem there. There's the other photosystem there. Here's the first electron transport chain there. The series of redox reactions. The energy is harvested by this system here, which is a proton pump. And it is pumping protons across into the interior of the thylakoid. So this interior of the thylakoid progressively accumulates more and more and more protons. That means it gets more and more acid, but there's a chemical difference and a charge difference. The chemical and charge difference is between the inside and the outside. And the only way for these protons, we've got protons which are being pumped from the outside, but we've also got protons which are arising from the splitting of water, don't forget. And those protons now can flow through this. This is ATP synthase. And that's exactly the same enzyme that we saw in cellular respiration, and it works the same way. It allows the proton charge to be equalized. It allows that chemiosmotic difference to be equalized across the two sides. And in doing so, it harvests the energy and it manufactures ATP. So the net products of this whole light gathering system is NADPH there and ATP there. And all of this is going to go off now to our next stage of photosynthesis, which is the Calvin cycle. So the ultimate product of the Calvin cycle is actually a sugar. So you think of glucose, which is C16 H12O6, and you'll see it fits that formula, CH2O, which is the formula for carbohydrates. So just remember C16H12O6, glucose, in your mind, because you may be a little bit baffled that the products of the Calvin cycle is not directly glucose, but I'll explain exactly how it works. So the Calvin cycle is the series of metabolic reactions in plants that takes place in the chloroplast. These are reactions which are not light dependent, but they are carbon fixing reactions. So the Calvin cycle is where carbon dioxide is fixed. It is put into a substance which is then passed through all of the metabolic reactions that comprise the Calvin cycle. And ultimately, the carbon dioxide is reduced, and we end up producing a substance called glyceraldehyde 3-phosphate. It is shortened to G3P, but please remember glyceraldehyde 3-phosphate. It's the one name I want you to remember. The Calvin cycle is very complex, and it actually has 3 phases to it. The first is the carbon fixation phase, which is accomplished by a very important substance and a very, very, very important enzyme, which I'll show you in a minute. Once the carbon, what happens is the carbon gets bound into a complex biomolecule. And that molecule is what goes through the whole cycle. And that molecule, now containing the carbon from the carbon dioxide, is reduced. But this is a cycle. So we have to regenerate the original carbon dioxide acceptor. And that indeed happens. So have a look. We're going to see that glyceraldehyde 3-phosphate has 3 carbons. Well, we're going to pull in carbon dioxide one at a time, and we're always going to be regenerating the carbon dioxide acceptor, the substance which bound the carbon dioxide. So in fact, we have to allow the cycle to run 3 times in order to produce 1 glyceraldehyde 3-phosphate. Carbon dioxide only has 1 carbon. We're making a 3-carbon compound, which is going to leave the cycle. We have to run the cycle 3 times for glyceraldehyde 3-phosphate. And that is going to be the product of the Calvin cycle. But the ultimate product is glucose, which is a 6-carbon compound. We take 2 G3Ps and we're going to join them together to make the glucose. So you need to remember, the cycle needs to run 3 times for G3P. It needs to run that twice to make 1 glucose. Okay, so it runs 6 times in all for each glucose molecule that is produced. Yeah, it's a bit difficult to follow, but let's just do it step by step. You do not need to remember the intermediates. Just be aware of the processes that we are watching happen. Okay, here is the carbon dioxide. This they are showing this having run 3 times to produce G3P, glyceraldehyde 3-phosphate. This substance here is a very important substance, quite a complex biomolecule called ribulose bisphosphate. And ribulose bisphosphate is important in biology because that is the substance which actually binds carbon dioxide. And it does it with the assistance of this enzyme. Some biologists call this the most important enzyme in biology. It is certainly one of the most abundant. And this is Rubisco. The proper name is ribulose bisphosphate. Yes, go ahead.
So that enzyme is used to speed up the process, right? This whole reaction.
Correct. Yes, this enzyme is what gets ribulose bisphosphate to bind carbon dioxide. So if you count this up, 1, 2, 3, 4, 5, it's a 5-carbon compound. When it binds carbon dioxide, it ends up being a 6-carbon. 1, 2, 3, 4, 5, 6. Okay. So Rubisco is called ribulose bisphosphate carboxylase oxidase. I know it's very long, but I've given you the name in another slide. Okay, so we'll get there. That's a name you must remember. Rubisco is ribulose bisphosphate carboxylase oxidase. And look, you remember that, and you can drop that into conversation somewhere and impress people. So we're going to start off with ribulose bisphosphate. Ribulose bisphosphate binds the carbon dioxide. It fixes carbon dioxide. Now there is a lengthy series of reactions. All of these are reduction reactions. They are reducing this very complicated substance. This, by the way, immediately splits into 2, 3 carbons. But what I want you to really observe, remember this is for 3 carbon dioxides entering, okay? This is per glyceraldehyde 3-phosphate molecule we're going to produce. Look how much energy it takes. 6 ATPs there to produce 1 molecule of G3P. 6 NADPH need to be oxidized to produce 1 molecule of G3P. Okay. This is all, these are all, what is happening here is we take this 6-carbon molecule here, we're going to, it breaks up into 2, 3 carbon molecules, and we end up releasing 1. For every 3 spins, we'll release 1 molecule of G3P. But there's something strange. Look at this. In actual fact, we produce more G3P than that, but we donate a whole lot of the G3P to regenerate the ribulose bisphosphate. Okay, we have to regenerate this. This is a cycle. If we don't regenerate this, the cycle stops. So this part of the G3P product is always donated in order to recreate the ribulose bisphosphate, and we use up more ATP. This is regeneration. So we have carbon fixation there, reduce it, regenerate the ribulose bisphosphate, and you're ready to go back to fix another carbon dioxide molecule. You have to run this 3 times to spit out 1 G3P, but don't forget, we need 2 G3Ps to make 1 glucose. So we have to run this whole thing all over again. The whole thing all over again to get another G3P to manufacture glucose. That's why I told you it is very, very expensive. In terms of using biologically useful energy and reducing power, it's a very expensive process. But don't forget, the energy is free. It doesn't matter because the energy is free. As long as the light is shining on the reaction centers, we've got an abundance of ATP, we've got an abundance of NADPH. As long as we have an abundance of those and we have an abundance of carbon dioxide and water, the process just proceeds. And it ends up producing compounds which are extremely rich in energy. They are carbon compounds which have been reduced with highly energized hydrogen electrons, hydrogen atoms. And there is a lot of energy bound into the bonds between the carbon and the hydrogens.
Professor?
And that, yes, go ahead.
This light, it doesn't have to be just sunlight, right? It could just be regular daylight.
Well, regular daylight is sunlight, but it can be any light. You can use an artificial light as well, as long as it's the right wavelength.
As long as it's present and has the right wavelengths, this
Correct.
process can get carried out because ATP is being produced.
Correct. It's producing both ATP and reducing power. And that's all that the cycle needs. Because the cycle runs, it regenerates its own ribulose bisphosphate. The enzyme sees to it that the carbon dioxide is fixed, and everything else just proceeds as long as we have an abundant source of ATP and reducing power, which we do, because this all comes from the light reactions. Okay. So as I say, let me just go back here. What I need you to, the things I really want you to remember, first of all, the product, which is glyceraldehyde 3-phosphate. And remember that 2 glyceraldehyde 3-phosphate make 1 glucose molecule. I want you to remember the carbon acceptor, which is ribulose bisphosphate, and the name of the enzyme that governs this whole reaction, which is ribulose bisphosphate carboxylase oxidase. You'll see it written down, and you can remember it from there. Okay, so the net output of these 3 turns of the Calvin cycle is 1 molecule glyceraldehyde 3-phosphate. Run it again, and you get another molecule of glyceraldehyde 3-phosphate. Put those are put together, and they form 1 molecule of glucose. So we think glucose is the product of all of the photosynthetic reactions. Remember that in plants, first of all, the plant is funding its own respiration. It's funding its own growth and everything else from what it produces from photosynthesis. But it often produces in abundance. It often produces glucose in abundance and in excess. And the plant will then take glucose, it will unite glucose molecules, it will polymerize them into starches in plants. And that is their primary storage product. But starch is nothing more than massive gazillion glucose molecules all joined together, sitting waiting to be utilized in producing biologically useful energy. And the plants do produce their ATP, apart from what is produced here in these light reactions. For the rest of it, plants behave exactly the same as every other eukaryote. Plants have mitochondria, which produce biologically useful energy in the form of ATP, and they do it by cellular respiration of the glucose that they have produced during photosynthesis. The plants photosynthesize and respire. They respire the whole time. They have to, because their cells have metabolism which is running exactly the same as any other eukaryote. It needs a constant source of biologically useful energy in the form of ATP. And plants provide that from their mitochondria. So plants have cellular respiration running the whole time. Photosynthesis operates in the light. Okay. Remember that don't think that plants don't respire because they photosynthesize. They do both at the same time. There's the full name for you, ribulose bisphosphate carboxylase oxidase. And remember, this is the enzyme that governs the reaction. The substance that accepts the carbon dioxide is ribulose bisphosphate. Now you fill this all out yourself in your revision. Fill this all out. How many terms of the cycle for G3P? How many for glucose? How many molecules of ATP are used? You can do it per glyceraldehyde 3-phosphate molecule or per glucose molecule. Just make it clear. How many molecules of NADPH? How many molecules of carbon dioxide? How many molecules of ribulose bisphosphate are used up? That is a trick question. Why? Because it's a cycle. So that needs a little bit of thought. So in talking about the importance of photosynthesis, I want to tell you a little bit about the origin of life on the planet as we understand it. We believe that life originated on the planet in a soup of organic molecules, or a sea full of organic molecules, which had been created by non-biological processes. You can look up in your book on the origin of life. We don't discuss it much, but there was a period when organic molecules were being produced from simple compounds, not by biological processes, but by non-biological processes like lightning, this sort of thing, volcanic activity. And life evolved in the soup. And very soon there was a crisis because living things were purely heterotrophic. They were perfectly happy living in the soup of molecules. They had all the molecules they needed to sustain life, but there was a crisis because soon they began to use up all of those organic molecules, and they broke those organic molecules down for fuel purposes, etc., to simple compounds, back to carbon dioxide and water. And this has been termed the first great extinction crisis, because living things which were prokaryotes at that stage were running out of food. And this system of photosynthesis appears to have evolved during that time. And the system of photosynthesis was one which evolved in prokaryotes to provide them with a source both of molecules, of organic molecules, and a source of energy. And once that was done, once that system evolved, then all biological systems were funded with both molecules and with energy, because that's ultimately where we derive all of our molecules. It's where we derive all of our energy, is from what the autotrophs do. Ultimately, we depend on the energy which the autotrophs fix, and the fact that they fix that into organic molecules, which we use as our source of molecules as heterotrophs. So that was extinction crisis number one, was when prokaryotes started to run out of food, and the system of autotrophy had to evolve in order for things to survive. But extinction crisis number two arose because of this system of manufacturing food from carbon dioxide and water using the sun's energy, because don't forget, the byproduct of photosynthesis, a waste product of photosynthesis, is oxygen. And this brought about extinction crisis number two for all living things, because very soon oxygen began to build up in the atmosphere. Up until that time, there had been no free oxygen at all. Any oxygen was bound up chemically to things like iron in the form of rust, that sort of thing. This system of photosynthesis, when it evolved in the prokaryotes, very efficiently produces oxygen. It produces masses of oxygen. It produces all of the oxygen that we have in our atmosphere today, which is a high proportion of the atmosphere. And unfortunately for biological systems, oxygen is extremely dangerous. Oxygen is a very reactive molecule. Not only is it reactive on its own, but it has reactive subspecies like oxygen singlets, just single oxygen atoms. It's usually in the form of two oxygen atoms joined together. But if you have one single one, that's called an oxygen singlet. It's very, very reactive. And you can have three joined together as ozone, and that is even more reactive. There are all sorts of permutations of oxygen which damage biological molecules. And this is extinction crisis number two. things that had no resistance to oxygen were killed off. And that, we believe, is where the system of cellular respiration first came into play. Cellular respiration, remember, is the exact opposite of photosynthesis. Cellular respiration takes organic molecules and it takes oxygen, free oxygen, and it uses the free oxygen to break up the organic molecules to extract energy, and ultimately it produces carbon dioxide and water, the starting point of photosynthesis. Once cellular respiration evolved, these two systems fit in together. Photosynthesis takes carbon dioxide, water, sunlight, makes organic molecules, which contain energy and the atoms that everything needs. Cellular respiration takes that and takes oxygen and renders it back to carbon dioxide and water and extracts the energy to make ATP and utilizes whatever atoms they need. So that globally, the biosphere runs by running photosynthesis and cellular respiration together. Okay, this is just a summary here. The light reactions, don't forget, and the Calvin cycle reactions. And I think that is that for this one. Okay, just hold on one sec. Yes, go ahead.
So
I'm listening.
I had a few questions while you changed the slide. First question I had was, are you adding the extra credit that we did to the actual test grade, or is that going to be separate?
Yes. No. Any extra credit, it gets added as an extra to your final grade.
And then
if you got 2% extra credit points and you earned 86, then your final score is 88.
On the test?
No, on your final grade.
Oh, on your final grade. Okay, I see.
Yes.
And then are you going to post that grade to let us know what we get, or are we going to?
Yes, I will let you know. Yes,
okay.
I'll let you know what you get. Yeah.
And then the next question I had was, are we having a quiz on Wednesday?
You will be having a quiz on Wednesday, yes. Just a short quiz, as usual. Just a few questions.
At 1:50, right? At 1:55 it starts?
Correct. Yes.
Okay. Good.
Yes.
Okay. Thank you.
Sure. Now, I hope this works. I hope I don't lose everybody or something. I just need to find my next set topic. Okay, here we go. Oh, man. Where am I? Technology and I don't get on all that well together. Where am I? There I am. Okay, here we go. Okay, can everybody see that?
Yes.
Okay.
Yeah, we can.
Thanks. I just want to try and get you all back. There we are. All right. So, well, we got to the point we've talked about how we build biomolecules, how we put them together into cells, how plants fund all the biology by photosynthesis, etc., etc., etc. Now we need to start talking about the one great property of life itself, and that is that living things reproduce themselves. Well, they reproduce themselves in two ways. Every cell in a living organism has to be able to reproduce itself. And in addition, the whole organism needs to be able to reproduce itself. Those are actually two different processes, but they are allied in that we have to consider how it is that we actually take a single cell and we get it to replicate itself and divide and do so accurately. The first process that we're going to talk about are the actual cellular mechanisms. We're going to watch how a cell physically does that and divides. And then later on, we're going to talk a bit about DNA structure and how DNA is replicated. But for the moment, what we're going to be considering is what we refer to as the cell cycle. And the cell cycle refers to the fact that periodically most cells will undergo a period of reproduction. And it depends on the purpose as to what type of cellular reproduction it is. But the most common form of cellular reproduction is where a cell actually divides to give rise to two identical daughter cells. It's a bit different when we talk about the reproduction of the organism. We'll talk about a different kind of cellular reproduction then. But the most common form of cellular reproduction is a form of reproduction where one cell divides and ends up producing daughter cells which are at least initially identical to one another. So we're going to start off our consideration with a parent cell. The parent cell will do whatever, it grow to a certain extent, whatever, and then there are very clear signals sent to the cell when the cell needs to begin dividing. But for most of the cell's life, it exists in its working form. And that we call interphase. And during interphase, that's when the cell grows. It increases in size and everything. And one really important event takes place as well. During interphase, the entire genome of the cell, all of its hereditary material, is replicated. The cell does not necessarily immediately start dividing, though. It may remain in this phase, which we call interphase, for a long time. And in actual fact, there are some cells in our body which remain in that interphase almost indefinitely. Our nerve cells, for example, they very seldom divide. But other cells of our body, actually their interphase is quite short. Our skin cells, for example, they reproduce frequently. So it just depends on the nature of the cell as to how long this interphase is in relation to the rest of the...
Professor?
Yes, go ahead.
All cells in the body carry genetic material?
All our cells do. All of our living cells carry identical genetic information. We all have all of our cells have exactly the same DNA. It's just it depends what genes have been activated, which have been silenced. That's what gives the cells their identity. But whether a cell has whatever genes are active in a particular cell, when the cell divides, the whole complement of DNA has to be replicated so that all cells inherit exactly the same DNA.
And then in interphase, when that genetic material is copied, it stays in that one cell. So when it's time for the next phase and the second cell is made, the genetic material automatically gets transferred to the second cell, right?
Correct. Yes. Yeah. We'll see the exact mechanism in a minute. I'm trying to get my little spotlight. I can't do it. Anyway, I'll just have to use the arrow. Yeah. There we go. Okay. So interphase is usually the longest phase. And that really, you can think about it as being the working phase. That's when the cell's doing all its normal business, is during interphase. But the cell has very particular mechanisms whereby it is informed about when it's going to need to start dividing. And once it does, then it'll enter into a division phase. the commonest form of division is this system where one cell divides and produces two identical daughter cells, and that we refer to as mitosis. Mitosis, and in mitosis, remember, we've duplicated the DNA here, and during mitosis, those DNA duplicates are separated one from the other and distributed now to the two new daughter cells. After that, there's a process called cytokinesis. Cytokinesis is actually the division of the cells in between. Mitosis is the separation of the genetic material. Cytokinesis is the actual division of the cells and their separation. I'll just mention a couple of things. First of all, in eukaryotes, the DNA is organized as a single length of DNA, single DNA molecules, and those constitute a chromosome. So this is a simple diagram here. They've shown you an organism here with one chromosome. We, of course, have many chromosomes. We have 46 chromosomes in all, 23 pairs of chromosomes. And but just for the purposes of this, they've shown you an organism with one chromosome. So here it is here. Initially, at the start of interphase, it's a single DNA molecule. During interphase, that DNA molecule replicates completely and identically. And the two molecules sit closely stuck together for the most of interphase. If we were to look, in fact, you can hardly, really, it's very difficult to distinguish that there are two molecules stuck together like this, but they are. Then in mitosis, we're going to separate these two identical copies, one to one cell and that one to the other. And we end up with these two identical cells, and they are at exactly the same place as this. They're at the start of interphase. So the one thing that you need to know, during this process of separation, during mitosis, almost all of the normal functions of the cell end. They have to be suspended because the DNA is not available. The DNA is not available for the genes to be read off and produce all their products and everything else. Most of the functions of the DNA shut down during mitosis. So it is essential that mitosis, that the actual separation take place very rapidly so that the cell can return to its normal activities as soon as possible. And it does, indeed, that is exactly what happens. So, this kind of cell division that we're talking about here, mitosis, is really important because it pops up all over the place. First of all, it's the way in which most single-celled organisms reproduce. Most things like paramecium, all of those, the protists, all of those things, they all reproduce asexually. As well, they may have some sexual reproduction as well, but their most important form of reproduction is asexual reproduction, where they divide by mitosis to produce two identical copies. Second of all, in multicellular organisms like ourselves, our development is all driven by mitosis. Think about it. We start as a single cell, as a single fertilized ovum. And that ovum, fertilized ovum, divides and divides and divides and divides by mitosis to produce the fetus and then ultimately the viable infant. During growth of the organism, from infancy to adulthood, all of that growth is funded by mitosis, by mitotic division of cells. Now, when they divide, the cells may go on to adopt new fates, but they all arise as identical copies of an original cell. What I mean by that is that we have many cells in our body. For example, our skin cells. We have a layer of skin cells which continually generate the cells of the epidermis, the outer layer of our skin. Those cells divide by mitosis. Initially they divide into two identical cells, but one cell remains in its original state. The other cell proceeds to change its metabolism, its fate, everything else. It develops into something else. But initially, it's a true mitotic division because the original cell, which you can think of as a stem cell, the original cell divides and produces an identical copy of itself. One of the daughter cells may go off to mature into something different, but initially they are identical. It's still true mitosis. The next thing is, skin cells are actually a really good example of continual mitotic division, which aids in renewal. Our skin cells are very short-lived. They continually die on the surface of our skin and are flaked off, and they are continually replaced by cells from mitosis underneath. But mitosis is also very important in healing. In all the healing processes, damaged cells are replaced by mitotic division of other cells. So mitosis plays a very, very important role throughout an organism's biology. Here's a protist busy dividing, an amoeba, and here the division is a simple division into two identical cells, each of which become completely independent. Remember, mitosis also funds growth and it funds development. And here, this I believe is a sea urchin, or busy dividing. Initially in division, even in our cells, our initial divisions, all the cells are identical. But as development proceeds, the cells begin to adopt different fates. They have different fates depending on their position in the embryo. And lastly, mitotic division is really important for tissue renewal, our skin, for example, and in repair processes. Okay, so let's just think for a minute about how DNA in particular is organized inside a cell. You'll often hear this term genome used, but actually I have to tell you it's usually used incorrectly. For instance, you will hear talk about the Human Genome Project, which is a scientific project to work out the DNA sequence of the entire human genome. But in fact, the Human Genome Project only looks at DNA, which is in the nucleus of our cells. Each cell's nucleus throughout our body has exactly the same DNA in it. No matter what the function of the cell, each cell has identical DNA. And it is conceivably possible that we can get the DNA sequence of all of those chromosomes. But the point I'm trying to make is that in fact, there is a small amount of DNA which is outside the nucleus. For example, in our mitochondria have their own DNA. And properly, the genome includes that. But loosely speaking, when people talk about the genome, they are usually referring to the DNA which is inside the nucleus of the cell. In prokaryotes, and things like the bacteria, it's a little bit different. The genome consists of a circular chromosome, a single circular chromosome, and then a number of other small circles of DNA as well, called the plasmids. There are some variations on the structure of chromosomes throughout the eukaryotes, but in general, in the eukaryotes, the chromosomes consist of single DNA molecules, as I described to you earlier. And in eukaryotes, each chromosome can be very, very, very long. Hundreds of thousands of DNA molecules, or hundreds of thousands of nucleotides, rather, all joined together in a single DNA molecule, and that may constitute anything from several hundred to several thousand genes. So let's have to start to think about what it looks like. This is actually a cell which has begun mitosis already. And I'm showing this as a cell to show you that at this stage here, we can see these thickened threads here, which is all, these are all chromosomes. These are all DNA molecules which are busy condensing down. They are getting thicker and thicker and thicker. What is actually happening is they are getting tight, more and more tightly wound in a very intricate process that weaves them into tighter and tighter and shorter lengths. So this is near the start of the whole mitosis procedure. For the most part of the cell's life, during interphase, the chromosomes are actually invisible because they are unwound. They are very, very thin. They're extremely long, and they fill the whole nucleus. If you look at it under the microscope, all you see is a kind of a grainy background. And that's what they will look like for most of the cell's life during interphase. It's just that grainy background. You can't actually distinguish individual chromosomes at all. It's only as mitosis approaches that the chromosomes begin to condense down like this and begin to look visible. So at this stage also, I just want to mention that there are the two kinds of division. By the way, that grainy background, when the chromosomes are unwound like that in the nucleus, that is referred to as chromatin. So chromatin is just unwound chromosome material. It actually consists of DNA together with the proteins, and the proteins are what actually wind it, are responsible for winding it. So there are two kinds of cells in our body. If an organism reproduces sexually, the first kind of cells are the bulk of the organism. Those are the normal cells. They're called the somatic cells. Somatic means body. And these are all the non-reproductive cells of the body, the skin, organs, etc., etc., etc. But then there are a relatively small number of cells which are specifically concerned with producing the gametes. And those cells are the germ cells. Sorry, I didn't put germ cell here, so please make a note. Gametes are produced by specialized cells called the germ cells. And the germ cells are distinguished because they have a different chromosome makeup. They are actually able to undergo a process called meiosis. And meiosis is a very different process to mitosis, and we'll discuss the differences. But products of meiosis are the gametes, and the gametes are produced by the germ cells. Yes, go ahead.
So this is, I know that a process called meiosis, which produces these, is the reproduction of gametes.
Correct.
Females, I know that aren't they already born with their eggs in their ovaries?
Yes, we'll learn about that a bit later on. Yes, there's a difference between males and females. Females are born, most of their eggs have undergone the process of meiosis already. In males, we produce sperm continuously.
Right. So when a woman is, pregnant with a child, then this is during the process while the baby is being, created and is developing inside of the uterus. This process happens as well. So
no,
meiosis.
No, meiosis precedes it because the egg has to have half the number of chromosomes. because it has to be fertilized by a sperm to get the full number of chromosomes. So it's essential that the egg only have 23 chromosomes, the sperm only have 23. Our normal complement is 46. So egg 23, sperm 23, together 46. We'll discuss it in more detail when we get to meiosis. We'll discuss the exact sequence of events, okay?
Okay.
But you are correct. You are correct. In fact, it's a quirk that in females, most of the processes of meiosis are actually completed when the female is actually herself in the uterus as a fetus. And then they sit and wait. They wait to mature and be released. But in males, the process of meiosis is continuous. We're continually producing gametes. Okay, the rest, all the normal body cells and everything, those are called the somatic cells, and they're the non-reproductive cells. It's somatic cells that we're going to be looking at first. And the key point to mitosis, is that during mitosis, we have to distribute the DNA equally. We actually have to take duplicated chromosomes, and we have to ensure that they are evenly distributed to each of the daughter cells. So when we look at the chromosomes, I'm going to flip back all the way back to here. When we look here, we start off with, this is a chromosome, and during interphase, it duplicated itself. This is actually what for much of the time, this is what chromosomes look like in the cell. And what you need to remember is that this is identical to this. Each of these is called a chromatid. The whole thing is still referred to as a chromosome, but it has two chromatids, each identical to one another. And during mitosis, we're going to watch as those chromatids, identical chromatids, get pulled apart from one another during the process of mitosis. Let me get back to where I was. So those two chromatids, once the DNA has replicated, they stay stuck together. They are sister chromatids and they're joined. But you need to remember something else. And that is that our chromosomes exist in pairs. We have 23 pairs of chromosomes in the human being, for example. The number of chromosomes varies enormously depending on what organism you're talking about. But in human beings, there are 23 pairs. And each pair, one chromosome comes from the mother, one chromosome comes from the father. One chromosome is maternal, one chromosome is paternal. They carry the same genes. So, for example, I might have a gene for hair color on the maternal chromosome. On the paternal chromosome, I would have a gene for hair color in exactly the same place. It's just that it might be for a different color. Okay, the genes are not necessarily identical, but their position on the chromosomes is. So you need to remember that all the way through when we're talking about mitotic division. Remember that we are talking about the full complement of chromosomes. In human beings, it would be 46. Let me go flip back here again. You see here, there's a slight narrowing there. There's actually an important place on the chromosomes there called the centromere. And the centromere is that waist, but it has some specific proteins whose purpose I'll tell you about in a while. And it's visible as a narrowing where those two chromatids are closely joined to one another. And we're going to watch the centromere during the process of division. So here we are. These are each sister chromatids, right? This one, absolutely identical to that one. Why is it identical? Because it was derived from that one. It was derived from the replication of this molecule of DNA. They are joined here at the centromeres. And these are the chromosome arms here. They refer to them as the arms, so they have a short arm and a long arm, depending. And we can recognize the chromosomes, each, like our 46, our 23 pairs, they have physical characteristics of size and of shape, which enables us to identify which one pair, how one pair is distinguished from another. And they are given numbers. We have pairs 1 to 23. Okay, so here's what's going. This is kind of a reprise of the simple diagram originally, because here we're going to watch the fate of one chromosome. Here's the chromosome in a little bit more detail now. Here's the centromere. Here are the chromosome arms. During interphase, this chromosome is replicated. It sits with its daughter replicate, and they're now called the chromatids. Closely stuck to one another, there they are there. And that is the classic X shape of the chromosomes. Notice what has happened. We started off with one molecule of DNA in the chromosome, making up the entire chromosome. We duplicated it entirely. So we've got two identical molecules. What we're going to watch during mitosis is the separation of those chromatids, one from the other. At the same time, remember that this chromosome has a matching. If this was maternal, it would have a matching paternal chromosome. And the same processes are going to happen to that chromosome. Okay, so here's to get this into your head as an initial difference between mitosis and meiosis. Mitosis is the assortment of identical genetic material to two daughter cells. And during the process of mitosis, we separate the chromatids. And we then divide the cells during cytokinesis. And we end up with two identical cells. And we also end up with a full complement of chromosomes. If it was a human cell, I start off with a human skin cell. It has 46 chromosomes in it. By the time the whole process is finished, I end up with two cells. Each of them has 46 chromosomes. They have the full complement of paternal and maternal chromosomes. We say that they are diploid because we've got 23 pairs, and the pairs are all complete. The nucleus is said to be diploid. We designate that as 2n, n being the number of pairs, and there are 2 in each pair. So there are 23 pairs times 2, 46 chromosomes altogether. The normal state, somatic cells are diploid. They are 2n. Gametes, however, are produced by the germ cells. And the germ cells undergo processes which lead to a sort of division, which is called meiosis. And now at the end of meiosis, instead of 2 identical cells, we end up with 4 non-identical cells. You'll see why they're non-identical in a while. It's a very fantastic process. But the gene complement of those 4 cells will have been cut in half. Each of the 4 cells will have half the number of normal chromosomes, 23, 23, 23, 23. Okay, so we say that they are haploid. They have half the number of normal chromosomes. They are haploid, or N. But here's the thing. We're using this to make gametes. And we're going to get one gamete, N, to fertilize another gamete, N. And we end up with a cell, which is diploid, 2n. That's the point of meiosis. The whole point of meiosis is to halve the amount of genetic material.
Professor?
All right. Yes, sir.
I'm going back, I think, like 2 slides. I just didn't finish copying like the last
That's
sentence.
fine. Which one?
One more back. Yep, that one. Just 1 second.
Okay.
Got it. Thank you so much.
It's okay. All right. So we're going to have a look now at the actual stages of mitosis. We realize that the cell undergoes a cycle. It undergoes a cycle of growth and then of division. And the division itself we refer to as the mitotic phase. And that is the whole process of division, of mitosis itself, and then of cytokinesis. And then the normal living period of the cell, which is interphase. And we also need to realize that within interphase, there are various processes taking place. It's as if a clock is running and the cell is moving through different stages as time passes. And the first stage we realize we can recognize is called G1 phase. As the first, it's called the first gap phase. The next phase is one of the most important phases. It's the phase where the cell replicates all of its DNA, and that's called the S phase, or the synthesis phase. The next is the second gap phase, and that's the period which immediately precedes mitosis. The cell grows during all of those phases, but it's only during this phase, S phase, that the cell replicates its DNA. Okay. Yeah.
So is this now interphase, prophase, metaphase, anaphase, telophase?
No.
No, not yet?
No. Nope. No, not at all. No. This is all to do with interphase. Here we are here.
Isn't interphase the longest phase?
Correct.
All of them?
Correct.
So like complex, there's so many things that go into it.
Correct. And its interphase is divided into these 3 phases. The first one, is G1 phase, gap phase, and that is just normal metabolic activity and growth. During S phase, the same processes continue, but at the same time, all the DNA is replicated. And then at the end of the S phase, G2 phase begins. The normal processes of the cell also do continue, but if you were to examine the cell closely, you would see that there are various processes taking place in preparation for mitosis. And I'll mention 1 or 2 of them. But there are distinct changes taking place in the cell here as it readies itself for mitosis, because it has to get all of the mechanisms ready. It's very difficult for the cell to have any of its genes operating and be producing proteins and things like that during mitosis. So they're all produced, everything, all the building blocks are put in place before mitosis. And then once the cue is given for mitosis to start, mitosis proceeds automatically and very, very rapidly. And we'll talk now about the different stages of mitosis. Okay, so interphase is most of the life of the cell. Mitosis occupies a very, very brief period of the cell's life deliberately. So we are going to recognize these following phases in mitosis. And just remember the names, we'll see them numerous times, so it'll get reinforced. Prophase, prometaphase, early metaphase, metaphase, anaphase, and telophase. Each of them absolutely distinct from one another. So let's have a look here. And I'm going to mention several characteristics. Try to make notes as you go along, because it's important to remember these things. If you need me to, I'll happily go over it all over again. Here is basically, you can see the different kind of preparatory steps. taking place in G2 of interphase. And in G2, one of the things that happens is that these little bodies here called the centrosomes here, they duplicate. It's one of the signs that mitosis is coming. Inside the nucleus, you wouldn't be able to see anything different because, in fact, the chromosomes are unwound and we say they're uncondensed, so you just see them as a background of chromatin here. As mitosis approaches, the first thing that you notice is that the nuclear membrane around the nucleus begins to break down. Mitosis cannot happen while there is a nuclear membrane. So the nuclear membrane disappears, and it disappears during prophase. Also, if you look in prophase, prophase goes on for a little while, but as you look as prophase proceeds, you'll see that the chromosomes begin to condense. They begin to shorten, get thicker and thicker, and ultimately by the end of prophase, they are visible in the nucleus. Have a look here very carefully, and you can see there and there. There are the duplicated centrosomes, one there and one there. And one of these is going to move around and it's going to come to this side here. The centrosomes are tremendously important. The centrosomes are the center of microtubule formation, and microtubules are essential to this whole process. I'll describe their working in more detail later on. For the moment, here's prophase. The nuclear membrane is breaking down, chromosomes are condensing, and the centrosomes are beginning to move. The next stage is early metaphase or prometaphase. And in prometaphase, here are the centrosomes here. And the centrosomes have spun what is referred to as the mitotic spindle. It's not very obvious here, but it consists of microtubules emerging from the centrosomes, and they go out and they join up to each of the chromosomes at the centromere. And have a look here and you'll see. Here's a microtubule and it's joined to the centromere there. Here's a microtubule joined to centromere there. Ultimately, they will be joined by microtubules on either side, there, for example, and there. And that's the responsibility of the centrosomes, is to spin these microtubules. And they grow them out until they contact the chromosomes, one on either side. So now each chromatid here has a microtubule attachment. In this stage, prometaphase, if you were to look at this in a living cell, you'd see all of these chromosomes starting to jiggle around. They're actually motile. They are mobile on these microtubules, and they begin to jostle around. They begin to move up and down and up and down and up and down. This is prometaphase, the early metaphase. In later metaphase, in full metaphase, those chromosomes all jiggle themselves right to the very equator of the cell there. This is a very important point. Here you can see the mitotic spindle quite clearly. And here are the chromosomes all being jostled around to get them to, till the centromeres are all on, the centromeres are all on this equator here. As the cell monitors that metaphase very closely, it monitors the structure of the metaphase spindle. And as soon as all of the chromosomes are exactly aligned along what we refer to as the metaphase plate on the equator, as soon as they hit that and they're all perfectly aligned, they split apart. And one chromatid goes one way and the other chromatid goes the other. And remember, this is identical to that. So here's another chromosome. It's chromatid, one chromatid that way, one chromatid that way, one chromatid that way. That separation we call anaphase. Once all of these end up distributed to the poles of the new cell, then the chromosomes begin to unwind and the nuclear membrane begins to form again. And it's at this stage now that the rest of the cell comes into play. And the rest of the cell begins to divide here in the process called cytokinesis, cell division. We have identical DNA on this side, identical DNA on that side, one centrosome on that side, one centrosome on that side. And now the nucleus begins to reform, the chromosomes unwind, and the cell now enters interphase again, and now begins to grow. And the clock again begins to tick. The metabolic clock begins to tick, waiting for the next mitosis. Okay, let's stop it there. If anybody has any questions, please feel free to ask.
So next class, we're going to continue with the rest of the steps, right?
Well, yeah, yes, we'll finish this and we'll do meiosis as well.
So everything we've covered today is going to be on the quiz, right?
Yes, we'll keep the quiz just to this class, only, yeah. Okay, anybody else? All right then, everyone. Then I'll see you on Wednesday. Wednesday will be a recorded class. But again, don't forget on Thursday, I have question time for you, which you're welcome to join if you wish to do so. And I'll send out a reminder of the meeting ID for that as well. Okay, so I'll see you then on Wednesday, or at least you'll see me.
Thank you so much, Professor.
Bye-bye.
Take care.
Yep, you too. Thank you very much. Bye.